of the Northeast Regional Floristic Quality Assessment Database Project. This project was funded by an FY 13-14 Wetland Program Development Grant from EPA Region 1, and we are very excited to see it completed. We'll follow this presentation with some time for questions and answers. If you'd like to ask a question, you can raise your hand or just send me a chat over to the chat box labeled Kimberly Roth. I'm your webinar moderator. My name is Kimberly Roth. I'm an environmental analyst with the New England Interstate Water Pollution Control Commission, or NEIWPCC. I manage our wetlands-related programs, including our Wetlands Work Group, Biological Assessment of Wetlands Work Group, and the Stream and Wetland Buffers Work Group. Please welcome Don Faber-Langendoen. He is a senior ecologist in the Conservation Science Division of NatureServe. He works with the Network of Natural Heritage Programs and Conservation Data Centers, and with conservation partners in the northeastern U.S. and eastern Canada. Don's key interests include the classification and conservation status of ecosystems, ecological integrity assessments, and monitoring methods. He has helped to develop NatureServe's international ecosystems classification with a special focus on the U.S. And in addition, through EPA support and other funding, he has helped develop and apply ecological integrity assessment methods to wetlands and coastal marine systems across the United States. All right, Don should be able to, oops, sorry guys. Okay, there we go. All right, Don, now take it away.
Right. Thanks, Kim. And thanks, everyone, for joining the webinar. NEIWPCC has not only provided funding for this work, but also given us a number of opportunities for us to present this project to New England wetland scientists. So as we conclude our work, we welcome this chance to present more widely. Additional support came through a technical advisory committee of state and federal agency staff in the Northeast, and they've helped inform and guide our work. And we thank them for their time and for Kim in keeping us all organized. We've been here before on FQA methods for the Northeast. And so I want to begin by acknowledging the state FQAs that were developed in 2011. That work greatly facilitated this work. And two people associated with that work were also part of our team, Don Cameron and Art Gilman. But that work also identified the problems with the FQA method that led to this project. And that work was summarized in a paper in the Northeast Natural History Conference, in which among the key findings were several, namely that differences in the C-value ratings among states suggests that a floristic quality assessment in one state will have a different meaning from the assessment in another state. Secondly, that findings further imply that state lists should not be combined into a composite COC for the region. Those findings are a problem if one of the goals of an assessment is to be able to compile and assess wetlands across regions, and that's one of the goals of EPA. And so that's what led to our work, which was certainly a parallel to many other efforts around the country to put C values on an ecoregional basis rather than on a strictly state basis. So our project goals here were to make the FQA approach a scientifically defensible and accessible methodology for the Northeast. Our goal is to develop C values for ecoregions because state values limit our understanding of species response to stressors. We want to validate those C values and to provide a accessible FQA database or website tool. So the outcome, we hope, is a validated FQA methodology with an ecoregional approach that will be applied throughout the Northeast region to assess site conditions. going to break the talk into 4 sections. I'll first briefly summarize the FQA methodology, then talk about 2 objectives together, that is developing the species list by ecoregion with the ecoregional C values for those species. And the third objective is to validate the FQA metrics that use those C values, and finally to talk about the database and web tools. So many of you are familiar with the Floristic Quality Assessment methodology, but I just wanted to go over the methodology briefly to set the stage for our work. So FQAs are focused on assessing the degree of naturalness or stressors of a site based on the individual species fidelity to specific habitats and anthropogenic stressors. You can think of plants as biological gauges that are out there in the wetlands and uplands. And just as we often think of plants as having behaviors associated with their tolerance for acid, neutral, or alkaline conditions, or some variation on that theme, or having wet, moderate to dry indicator status, so we can also think about them as having indicator status for the degree to which they're sensitive to anthropogenic stressors. The methodology for assigning Floristic quality values was initially developed by Swink and Wilhelm, and Wilhelm is still very active, a constant presence in many of these kinds of meetings. And FQAs are now widely available for many states, as well as regions. And here in the east, the southeast, mid-Atlantic, and now northeast have ecoregionally based C values. So the core of the methodology then is that plants are weighted, individual plant species are weighted based on the breadth of their response to ecological and anthropogenic stressors. This weighting factor we call the Coefficient of Conservatism, or C value, and it ranges from 0 to 10, 0 being assigned in our case to all the non-natives, which have a very broad ecological tolerance, to native plants with a broad ecological tolerance given a value of 1, all the way up to native plants with a very narrow ecological tolerance of 10. And those C values then can be assigned have often been assigned at a statewide level, but are increasingly also assigned at an ecoregional level. So to give some guidance to experts who assign these C values, there is text that guides what a 0 means, what a 1 or 2 on up to 10 means. So for example, 1 to 2 is a native invasive or widespread native that is not typically found in a particular plant community type. The common yarrow is a good example of that, where it's often found in weedy habitats. It's not particularly associated with any native plant community. Up to plants that have a moderate level of specificity to native plant communities, but still are tolerant of anthropogenic disturbances, such as marsh marigold, to those plants, species that are almost entirely confined to a few native plant communities and are not tolerant of disturbances to those communities. So that's the basic thinking behind the C values. Those C values then are used to calculate various metrics. I'm only going to talk about those metrics that we focused on for this project that are most relevant to wetland assessments, and that's the mean C and cover-weighted mean C. These are very straightforward metrics. The mean C simply takes the average of all the C values for a species at a site and uses that average as a gauge for the wetland condition. A slight modification of that mean C is to collect the cover value of each species and then to weight the mean C value by that cover. These metrics have been widely used in wetland assessments, and so we focus on them here as part of our study. Just to give an example of what that looks like, and I thank Christy Giannopoulos for this slide. On the left, you have a site where you have 50 species, and 5 have a C value of 1, 2, 3, all the way up to 10. And so if you simply would take the average C value of those 50 species, you'd get a mean C of 5.5. If you go to a different site, you'll see now that the red bar indicates that there are some invasives present, and so they get scored as 0, and we start seeing a decline in the number of high C-value species. That begins to drop the mean down to 3.6 in this case. And further, as invasives increase and the number of species with high C-values further declines, the mean C-value again further drops to 2.3. When we look at what's driving that, we often see that there is a degradation disturbance gradient that's associated with those changing C-values. And so we found that these metrics based on the C-values are informative of the amount of stress or degradation that a wetland is experiencing. So that's the basic methodology that is driving our work. Again, our goal is to validate this methodology using an ecoregional approach that we can apply throughout the Northeast. So to do that, again, I'll focus first on these first pair of objectives, developing a species list by ecoregion with ecoregional C-values. The team we assembled for this, Ken Metzler, Don Cameron, Rich Ring, Art Gilman, and Leslie Snedden, who handled the actual ecoregional C-value assignments. They each had a primary ecoregion that they were responsible for, and we worked collaboratively to standardize our assessment of species across ecoregions. Mary Harkness and Kristen Snow from NatureServe provided the database support for handling all of our C-value estimates. So the ecoregions we're responsible for are the 5 ecoregions that cover EPA Region 1, and then with New York also added in because many of the ecoregions in those 6 states extend well into New York. So the way in which we went about assigning the C-values, we first compiled the state C-values that I mentioned earlier on that were done in 2011. And so we have for each species in the Northeast already a state C-value available to us. We then standardized that taxonomy of each of those state lists to USDA Plans 2017. We then created an initial ecoregional plant species list, and we relied strongly on USDA Plans county information for that. We then also calculate an initial ecoregional C-value using the state C-values weighted by their area of the state in the ecoregion. And I'll go over steps 3 and 4 again. Based on those initial species list and ecoregional C-values, we then got together as a team. And we first focused on review within ecoregions, then across ecoregions, and then clean things up. It all sounds so simple when I say it this way. Let me just go back over steps 3 and 4. So how do we get that initial ecoregional plant species list? Well, working with USDA plans, we overlay the county distributions available to us from USDA plans. Those county distributions are not complete, but as you can see from this map, there are many counties within an ecoregion. And so we were able to use that list to get, that distribution information to get an initial list for the entire ecoregion. Whenever a species occurred in any single county within the ecoregion, we gave it a yes for that ecoregion. If its only reason for occurring, for being listed in a county was when that county was split between 2 ecoregions and there were no other counties that were entirely within the ecoregion, we gave it a maybe, otherwise we gave it a no. Most of our expert review was focused on the no's, and we tended to accept the maybes as a yes as far as doing our ecoregional screening work. So that's how we got the ecoregional lists together. Then the part that relied on the state list. So this is a bit of a calculated project that then went through expert review. So what we have is, what's shown on the screen is what we did for one of the ecoregions. We did this for all of them, but this shows one of them, which is Ecoregion 58. As you can see, when you look at the Ecoregion 58, which is the mountainous region that covers New England, you can see that, for example, Connecticut has a very small part of the ecoregion. Massachusetts also has a small part. And the bulk of the ecoregion occurs in Maine and Vermont, with also large parts in New Hampshire and New York. And there are no parts of that ecoregion in Rhode Island. So when we look at Ecoregion 58, and we have a species such as balsam fir, we will have, therefore, state values assigned to that species by each of those state FQA products. So in the case of Connecticut, it was assigned a very high value of 8. In the case of Massachusetts, 7, Maine 3, New York 3, New Hampshire 3, and Vermont 3. What we did then was to weight each of these C values by the percentage of the state that occurred in the ecoregion. So even though Connecticut has a value of 8, because it only has 2% of the ecoregion, it receives a relatively low weighted contribution to the potential average, whereas Vermont and Maine, which have large parts of the ecoregion, carry more weight. So if you look across these scores, you can see that the weighted average of all these state COC values for that ecoregion is 4.2, which you rounded off to 4. Then we asked each of the ecoregional leads to review each of the assigned calculated averages to see if they agreed. If they agreed, they simply said yes, and we accepted it. If they disagreed, we'd expect a comment as to why they thought the calculated estimate was wrong. So that's for one ecoregion. We did this then for all the ecoregions with each of the leads looking at the weighted average assigned to each of their species. So from that, as you can see in this case, Ecoregion 58, balsam fir, had an accepted COC of 4 within the ecoregion. When you look across what the other leads did, okay, we've got 4 as the C value for Ecoregion 58. We have a 6 for Ecoregion 59, 3 for 82, and 5 for Ecoregion 83. So essentially, this process first generated an ecoregional estimate for the C value, followed by expert review, and then that led to a preliminary set of ecoregional C values for all species. Note that in this case, we also looked at the COC range. So in this case, the range of C values goes from 3 in Acadian part of Maine to a 6 in lower New England. In this case, the range of CC values is 3. We then got together as a happy group and crunched through all species that had an ecoregional range of 3 or more. And I'll show a table of that summarizing that work shortly. This shot is at the end of our meeting, and so we're looking a little giddy and happy about being done. But essentially, in 2.5 days, we reviewed all the species that had ecoregional ranges of 3 or more. That would seem to be where the ecoregional scoring had the most merit. So looking at this summary table, before the workshop, based on the calculated and expert evaluation of the ecoregional C values, we had from an initial list of 3,697 species, of which 1,172 were exotic, leaving about over 2,500 species that were native to the region. We see that many species had no variation in their C values across ecoregions, all the way up to 4 that had ecoregional values that ranged from either 1 to 9 or 2 to 10, generating a range of 8. So we focused again on those species that had ranges of 3 to 8, and so that was 552 species. So the group that got together, in this case in the Albany Pine Barrens, we looked at those 500 plus species in detail to see whether that ecoregional range made sense. At the end of the day, we resolved many of the differences downward so that we ended up with only 144 species that had an ecoregional range of 3 or more, which is about 5%. When you add in those with a range of 3 or less, it's down to 1%. So there's a relatively few set of species that have a wide range of C-values based on this process. So we then went through some post-meeting cleanup, checking with the Technical Advisory Committee to look over things. We did some further internal review, and that ultimately led to a set of ecoregional C-values for each of those 5 ecoregions, which are now available on the FQA, Universal FQA website, and they're also loaded into an EcoObs database, both of which I'll talk about later. So that met our objective 1 and 2. We have a species list by ecoregion, and we have an ecoregional C-value assigned to each of those species. Our third objective then was to do an FQA metric validation. Now, just as background again on the FQA methodology, the methodology is based on 2 key assertions, namely that there are areas with species assemblages that have greater fidelity to reference standard conditions, and those that fidelity reflects high-quality native habitat. And also, individual plant species differ in how they tolerate or respond to human or environmental stressors. It's this combination of fidelity to reference standard conditions and degree of tolerance to stressors that is at the core of C-values, as I've talked about earlier. Now, when I first thought about the validation process, we were thinking about this validating these general assertions. But as we progressed into the validation, I realized that although that's important, there's actually a more an extended question we could ask, which is, did our ecoregional C-value approach improve our ability to make these assertions? That is, did the C-values get more responsive to assessing levels of fidelity and degradation? And so we ended up wording our validation analysis in 2 ways. One is, do the FQA metrics based on ecoregional C-values instead of state D-values actually improve our interpretation of wetland condition? Well, how could we do that? If you look at the graph on the right, you'll see a standard comparison of an assessed range in conditions. In this case, lower condition or more degraded is 6, and higher ranges are 10. And then the mean C-value, again, the higher C-values mean better condition. And so you'd expect regression to look as it does, with higher mean C-values reflecting better condition. If we've done a better job with, or an improved job with our ecoregional C-values, we might expect the regression to get more highly correlated, and perhaps that the slope itself is steeper. That is, you get a better distinction between lower quality and higher quality sites. So we could do that. The other validation we took was based on literature review, particularly from Michael Bordash, who's worked in Minnesota on this quite extensively, looking at how much FQA metric scores vary by wetland type. And so we had enough data that we could identify where we could actually also test this assertion. And so this box plot is from Michael's work, showing that floodplain forests have a different score for high quality sites than do, for example, freshwater wet meadows. So even though these are both considered high quality sites, the C values for them are different. So we wanted to ask the same question in the Northeast. So the validation team, again, a different set of players here. with the Maine Natural Areas Program, Justin Slowan and Kristen Purrier were willing to help us with this, and so they actually compiled a comparison of their data sets and did a comparison of state C values to ecoregional C values. In Vermont, Everett Marshall and Eric Sorensen provided their plot data, which is pretty extensive, on a wide variety of wetlands, and we imported into the EcoObs database so that we could do the calculations for them. Charlie Hone, who was with the Vermont Department of Environmental Conservation, was chief instigator on this, and then he kept talking about the data set that Eric and Everett had, and we finally got around to following up his lead on that. And then Michael Lee helped with the transfer of the Vermont data into EcoObs, and Patrick McIntyre helped with a lot of the stats analyses. Now, we made this data set, or this validation exercise, a little bit harder than I perhaps intended, in that we restricted our data set analysis to within state. So as we've been talking about, a major goal of this project is to make sure that we can assess across state lines using the ecoregional approach. But our validation test actually was fairly fine-tuned that we said even within a state that has multiple ecoregions, can we detect an improved discrimination of wetland condition using ecoregional values rather than state values? When I realized how narrowly focused this question was, it gave me some pause, and I had to squint when various emails came through with completed analyses, fearing that we would not maybe turn out, that things would not turn out as well as we'd hoped. But you'll see. So our data set was largely based on 400 meter square plots that both the Maine and Vermont programs use, with full species lists that had cover values associated with them. So there were two data sets. The main data set had 158 plots, which spanned the gradient of wetland condition from A to C ranked. We'll talk more about those rankings, both for floodplain forests and for northern swamps. The Vermont data set was more extensive, but spanned a wider range of types, and was more narrowly focused on the reference condition, that is, the A or B ranked sites. So on the one hand, we have the ability to calculate these metrics, these mean C and cover-weighted mean C values from these plot data for these wetlands. On the other hand, the benefit of this data set is that each of these programs independently has been rating these wetlands based on the NatureServe network methodology we refer to as element occurrence ranking, or EO ranking. EO ranks are designed to provide an assessment of the estimated ecological integrity and probability of persistence of an occurrence based on an assessment of its condition, its size, and its landscape context. So looking at a particular wetland site, ecologists are evaluating the vegetation structure and composition, the hydrology, any soil alterations, as well as how big the wetland is and how well buffered and connected it is to adjacent natural areas. Those are put together into an overall wetland condition or EO rank assessment. And we typically have assigned, use a scorecard approach of an A, B, C, D rating, where A is excellent and D is poor. The state program staff had assigned EO ranks to all these sites, so they provide the independent scoring of level of degradation. So, looking at our FQA metrics, are the ecoregional C values an improvement over state C values? And this is where I could stop squinting at the screen, because indeed they are. As you can see here, the old scores are shown in blue, the new scores are shown in red, and the explanatory variables there are improved from 0.19 to 0.26. And the regression is slightly steeper with the new data, and not pretty modest, but it is somewhat of an improvement. So that suggests that our ecoregional scores within Maine are already an improvement over their state C values. You'll also notice there's a tendency for the new values to be slightly higher overall than the old scores. So we were perhaps more lenient or perhaps more faithful to the ecological behavior by looking at this in terms of ecoregions. So that was our first test, looking with main data where we had a larger gradient of high quality to lower quality. In this case, we were able to convert the main grades of A through C into a more refined score from 4 down to 1 because they have a scoring system based on additional metrics that allowed us to add finer divisions within the A through C. When we break this apart into wetland types, you can see the same pattern emerges, but one obvious difference is that the larger the floodplains, the correlations there are much less strong than are the correlations for the northern swamps. Now, there could be good ecological reasons for that. Floodplains are far more dynamic, and so the native species are maybe more generalist by default. So again, this may point to where a high score for one type may not equate to a high score for another, or even that the nature of the thresholds from A down to C might differ, and we'll come back to that. When we look at this in a more categorical form in terms of going with the broad ratings of A rank, B rank, and C rank, we can see again for Maine that now using box plots and looking at whether or not these means are significantly different from each other, we can see that it's possible to distinguish the A's and B's from the C's successfully with the main data. Note that there are very few plots that are ranked A, so that may have weakened our ability to pick up a difference with the A versus B. Conversely, now with the Vermont data, where we have a lot of reference sites that are A or B, we can distinguish A from B, but the number of C rank plots is low, and so that distinction did not show up on the Vermont side. But essentially, then, these metrics, the weighted mean C values, are corresponding or show correspondence to these broad levels of degradation or changes in condition. Now, this focuses on the cover weighted mean C, but just point out that it is worth looking at other metrics. Here, for example, is the mean C, and here you can see that for the main data, in fact, we can successfully distinguish A from B from C sites using that metric. Again, you can see how the average means for A, B, and C are somewhat higher on the with the new ecoregional C values. And we don't show it here, but if you look at the amount of variation that's explained by these box plots, there is, again, a slightly improved level of variation explained by the new C values compared to the old state values. So we're happy with that validation analysis. As I say, it is a fairly narrow test in that we're looking at within a state. A more broad test would bring together multiple data sets across state lines, within and across ecoregions, to see if there is an improvement there. Our second question then was the one of, do FQA metric scores vary by wetland type? We had, in this case, a more substantial data set from Vermont, crossing those 5 types, 4 of them wetland, 1 of them in upland. You can see clearly here that between the hardwood swamps and softwood swamps, there's a difference. Between those two and peatlands, there is a substantial difference. So fens and bogs have a much higher expectation for high C values when they're in good condition than do hardwood and softwood swamps. So again, the community type matters when thinking about thresholds for what determines higher quality mean C values versus lower quality. In the case of Maine, because we have the full gradient, we can also look again at whether it's possible to start setting thresholds for A versus B versus C rank sites. Here we just show the A versus C. And you can see again with the with the floodplain data where there's much more variability across the gradient, it's harder, though possible, to separate A from C in terms of thresholds, whereas in the case of northern swamps, there's a very clear set of thresholds one could identify for A-ranked versus C-ranked sites. So that points to the promise of developing mean C metric thresholds by community type. So just to conclude on that, the FQA metrics that are based on either state or ecoregional C values do show a positive response to a wetland condition gradient. So regardless of the fine-tuning or improvements, they both show good responses to the condition gradient. But there is a minor to moderate improvement, even within a state, over of EC values versus the state C values. We'd like to do further testing at a wider regional level. And we're now in a position to do that. But we also have to think about standardizing wetland types between states to allow us to properly calibrate those metrics. Finally, then, the regional database and web tools that we developed to support this project. Again, we had a website and database team. Will Freeman, who is the original developer of universalfqa.org, was very happy to collaborate with us to improve it based on the needs we identified out of this project. Michelle Boteau is our own programmer within NatureServe, and she and Will did all the design work for us based on input that Kristen Snow and I provided based on our suggested needs for making this a more ecological and wetland tool. Kristen and Mary and Michael Lee together worked on improving EcoObs as well to incorporate these ecoregional C value scores and metrics. So universalfqa.org, if you haven't been on that website, I highly recommend it. It is a very widely used tool for storing FQA databases and then for users to upload site data, plot data, translate data, whatever floristic list they have, and to use that website to then generate the FQA metrics of choice. I'm not going to go into detail about the website per se. I just want to summarize some of the things we did to enhance it for wetland use. It can now better handle plot data in addition to transect data. So there's now options for uploading, putting in data for a full plot as well as for subplots. And anyone who's done quadrite work within plots recognizes that there's always a desire to add in the rest of the species that are outside of the subplots but inside the plot, so we're able to tag those species separately from those that are within the subplots, plus identify associated species outside the plot that are relevant to the work. So some wetland scientists or ecologists like to put in a plot but then wander through the rest of the site adding additional plots, and so that data can be managed on the site. And you're able to then add or subtract in these various components of the species list and determine how that even affects FQA calculations. It's a little clunky because it's more or less added on to the existing functionality, so there are ways we could make that more streamlined. There's also now a wider choice of cover methods, and in fact the cover method calculations are now working properly. They weren't actually quite set up correctly before. There's a new sampling design section. There's new fields, both for the database side where you can now list the states, provinces, countries, as well as ecoregions that the database applies to. Though that's only for new databases going in. Old databases, there's a project there for someone to go in and actually catalog, to fill out those fields. There's also new fields for identifying the community type, more environmental description information, as well as an updated help screen. So one example of how this has been enhanced is it used to just have a transect assessment box. Now it clearly identifies this as allowing for transects and plots. There are now places to log in the community name as well as add notes about the community type. And then there's more information about what kind of transect or plot design was used to collect the data. I'm sorry for the poor details on this particular slide. It was a late thought, but I wanted to show you that, in fact, you can find the 5 ecoregional FQA lists on the website now. I also wanted to note that there is an additional FQA list from Kathleen Walter's team for New Jersey that was also released in 2017. And so right next door to our project, there's new information available to us from her work. Just to zoom in on each individual FQA project database from our project, this is the Northern Apps Highlands Ecoregion. Sorry, we used the North American coding here. We should add in the U.S. coding, which would be Ecoregion 58 that we've been looking at. And so it tells you the name of the database, the year it was uploaded, and then it gives you a brief project identifier. And you can view it online or you can download it and make it suitable for your own purposes. So all the information from this project is now available on the website. Then the second database or tool we have is a database that NatureServe developed called EcoObs. It is an in-house database, requires working with our staff to use it, but it is functioning across the U.S. as a database for anyone to put plot data and other data in. Its primary function is to allow users to put in vegetation plots and to do ecological integrity assessments. That said, it is set up more generally as an observations database. That is, you don't have to be doing plots. It could be any site list of species or ecological observations you want to make, and that can be managed in the site or in the database. You can also assign observations or plots to projects. You can then score them, do FQA calculations. You can import and export the data, and you can even upload the data to VegBank, which is a national repository of plot data. Importantly, too, there's a plants synonym lookup function. So although you can put in any plant species authority that you want to use for your plots, we also want to have a synonymy put in to USDA plants so that all plot data can be compiled and used together through a standard plant taxonomy. Just to take a peek inside the database, here is the most general tab showing the many fields that are available for describing the location of the observation. I'm not going to go into details, but I just wanted to highlight the overall set of tabs that are available for cataloging observations. And so those tabs span from the information about the classification of an observation to looking at very narrative observations describing the landscape, the site conditions, et cetera, to then moving through looking at vegetation, environment, soils, the kind of FQA metrics, other kinds of metrics that are used to score wetlands. And that together allows users to compile a good deal of information that describes wetland condition. Just to focus directly on the information used for FQAs, the database has a vegetation tab that allows you to describe the vegetation in terms of the overall structure, tree layer, shrub layer, herb layer, et cetera, as well as catalog individual species by those layers, assign them cover values using a variety of cover classes. And that's what then feeds into calculation of the floristic quality. You can then filter the observations, so if you have multiple projects, you can select the ones you're interested in. You can generate an overall cover value from the data if one is not available. And you can choose the FQA species list appropriate for your data, QC the data, and ultimately generate the same kinds of FQA metrics that are available through the website. But what EcoObs provides is a strong data management tool for all plot data beyond simply calculating FQA metrics. So that's the database. So those were the objectives we had for this project, and we have completed them all. Final report is in draft form and going to WPCC tomorrow, and then we'll be out for review for the next month or so before we wrap it up in March. Now, there are some next steps that we can see coming out of this work. One is to continue improving the tools we have. Certainly there's a need to continue testing FQA for wetland assessments, and Charlie Hohn's been talking about doing that with the Vermont RAM to look at how FQA scores relate to the RAM scores as well as to EORank from the heritage programs. There are further enhancements to the Universal FQA website. I think this becomes a user-dependent thing where the more that users are looking for wetland assessment kinds of applications based on plot data, the more there will be a need to improve the kind of functionality that are typically available for assigning FQA scores to plot data. We would like to extend EcoObs as a regional database tool. We continue to improve it through projects. We would like to make it more widely available. Ecoregional FQA lists need to be kept up to date, so that's a challenge. We also want to complete the ecoregional C values between the Northeast region and the Mid-Atlantic regions. As I noted at the outset, New York was only covered in this project through the ecoregion that extended into New England, but Ecoregion 60, which spans into Pennsylvania, has not been assessed, but the Mid-Atlantic region has developed regional C values, and so those values are available for comparison. And then, as I mentioned, Kathleen's completed the New Jersey state C values, so we could use the same approach for bringing that data in as we did for the Northeast, where we could look at the state C values for Ecoregion 84 in New Jersey and link that to our own assessment from Long Island and Cape Cod. That, by the way, would also give us pretty much a set of ecoregional scores from Maine down to Georgia. But beyond the actual tool development, there are applications. These tools are working towards allowing us to build reference data sets for wetlands. We have benchmark sites that can guide restoration mitigation. So we know where we can go to find where high-quality sites are with a full complement of high-quality species, reflecting what might be standards for restoration mitigation. Though these benchmark sites could also range down to good or fair sites so that restoration mitigation sites have targets that reflect achievable goals for sites that may not be benchmark standard but are still quality enough to serve as reference for those restoration mitigation projects. These reference data sets can also guide state or regional intensification of wetland assessments. And we also would like to see applications where we begin to build regional wetland classifications so that we can start setting thresholds for scoring these FQA metrics by wetland type. There are a number of existing regional classifications available. The state natural community classifications have been integrated into a U.S. National Vegetation Classification. It's available on usnvc.org. And the HGM classification is also available. It typically needs to be downscaled, if you will, to within regions to make it a little more sensitive to regional variation. It has been done, for example, in some of the mid-Atlantic states. And so those classifications can help organize the wetlands into regional types that will serve as better ways to aggregate the wetland data. So those are a few steps, and they will eventually allow us to have these reference networks available in the region that can guide a number of applications, whether it's assessments or restoration mitigation projects. So thank you very much for your time. Appreciate your listening in, and we'll pause now for any questions.
Great, thank you, Don. Great presentation. So I don't see any questions in the chat box. We can pause a moment, or you can raise your hand if you think you are interested in asking a question. Otherwise, we can...
Yeah, there was a bit put on through the whole presentation, but if there are any questions, let me know.
Okay. I'm seeing questions being sent over to the NEIWPCC Moderator box. I'm not really able to access them. Otherwise, you won't be able to see the screen anymore. So if you could please send them to Kimberly Ross up in the panelist box. Here we go. So we've got a question from Kathleen Wall. Thank you for joining us, Kathleen. She is wondering how to integrate New Jersey into the New England and Mid-Atlantic Ecoregional FQA.
Yeah, that's a good question, and probably one that we should sit down with EPA's Region 1 and 2 to talk through. I think the methodology for doing it is all there coming out of this project, and it's a matter of finding the funding support to pull a team together and work through the same database tools that we used with the expert review. And ideally, well, I know that Kathleen, you're sitting on plot data, so it would be, we would have a validation data set that would also be ready to go to test the outcome. So,
are there any changes to the universal FQA completed for all the states?
I'm not sure what's meant by the question.
Let me reread that. Are the changes to universal FQA completed for all the states? So
working with Will Freeman, he made sure that the changes to the website didn't affect any of the existing information in there. So there's been no changes to the existing FQA databases. It's only in the way in which data coming in to the website are changed.
Great. When developing composite p-values for each species, did you see a greater range for facultative species compared to obligate or vice versa?
We did not look at that. So that's an interesting question. Which reminds me that it was tempting to look under the hood and try to see what's going on between the old state p-values versus the new ones, and there's sort of a wealth of plant behavior strategies one might look at as to why they changed, but we didn't go there. But it would be interesting to look at what's driving the differences.
Is the NWCA plant data accessible in the universal FQA?
I don't know. That's a good question for Greg Serenbetz, I guess, as to whether or not that's been put on that website. It has a pretty good search function, so if it's tagged as NWCA in any way, then you would find it pretty quickly. I have not looked.
Is this approach valuable for assessing differences in wetland habitat quality on a state basis?
Yeah, I think there's a number of studies now that have used FQA values at least as one component of doing the wetland assessment. There is no silver bullet for wetland assessments, and I don't think FQAs necessarily stand by themselves. What they do bring to the table is a fairly rigorous, quantitative scoring, and I think in addition, what they provide is a clear sense of which species are really important to the community in terms of its quality. So it gives you that level of detail, which can be very helpful for guiding mitigation and restoration efforts. So there's a wide variety of literature available. I can certainly point people to some sources if they're looking for that on using these kinds of metrics for us doing statewide assessments or state-level assessments. And of course, I should add too that EPA used these FQA metrics, or used one FQA metric as part of its overall vegetation multimetric index. And so several of these C-value parameters were used as part of the national EPA wetland assessment.
Very good. Well, that looks like all the questions we received. So thank you again to Don for your work and your colleagues at Acres Reserve and all of the people on the TAC and all of the botanists that were hired to work on this project. We're really pleased to see it coming to a conclusion. So thank you for your time today and all the presentations you've done for us, and thank you to everybody who joined.